Hello everyone, welcome back to Dr. K. PREM-PRIMER video lecture series presenting by Dr. K. Prem, that's me. In today's lecture, we will talk about how to clone PCR products amplified by the Taq DNA polymerase. You see, PCR products amplified by the Taq DNA polymerase can be cloned by TA cloning vector. TA cloning vector. So this procedure is very specific to the PCR products amplified by the Taq DNA polymerase, but not any other thermostable DNA polymerases. So before going into the procedure, we will look into the different activities of Taq DNA polymerase. So Taq DNA polymerase is a DNA polymerase which can use the DNA as a template and produce the template. That's a DNA dependent polymerase, but it is a thermostable DNA polymerase which activity remains at a higher temperature. So it is having 832 amino acid and molecular weight is 94 kD. If you look into the domains and their activities, it is having two primary activities. One is DNA polymerase activity, that's in the C terminal, and 5' to 3' exonuclease activity, that's at N terminal. But here, in today's procedure, these two activities we are not talking. In addition to these two activities, Taq DNA polymerase have another associated activity is or a secondary activity is called terminal transferase activity. Terminal transferase activity. That means it adds a single deoxyadenosine to the 3' ends of double-strand DNA. During the PCR, Taq DNA polymerase, the PCR products which are amplified by the Taq DNA polymerase will have a single deoxyadenosine at 3' ends. That is due to the terminal transferase activity of Taq DNA polymerase. That's a secondary activity. So by using this activity, Taq DNA polymerase adds single deoxyadenosine to the 3' ends of the double-strand DNA. So basically, in a TA cloning vector, the insert or the PCR products have a 3' adenosine and vector will have a 3' T's. So vector with the T and insert with the A, they're complementary. So that's why it is called as TA cloning vector. We will see the procedure first, then we will see the method. First we'll see the text and followed by the cartoon. You see, to clone the PCR products, to clone a PCR amplificans, we have another approach called TA cloning, which creates a complementary single-stranded overhangs between the insert and vector by exploiting a secondary enzymatic property of Taq DNA polymerase. The Taq DNA polymerase has terminal transferase activity, which means it adds a single deoxyadenosine to the 3' ends of double-strand DNA during the polymerase chain reaction. That's a PCR. This unique property is only with the Taq DNA polymerase, not with any other thermostable polymerases. So here we are prepared the insert, that's a PCR products or PCR amplicons with the 3' overhangs, that's a single nucleotide A, And now we are going to prepare the vector with the T orangs. Two T orangs in the vector can be generated by first cutting the vector with the blunt-end restriction enzyme, and then by mixing it with the terminal deoxynucleotide transfer enzyme and dideoxythymidine triphosphate. It is missing the 3' hydroxyl group. The missing hydroxyl ensures that only single or only one T is added to the vectors. Otherwise, vectors can be digested with a restriction enzyme which generates the single T overhang. Single T overhang. Here you see, these are the PCR products. During the PCR, Taq DNA polymerase can add single deoxyadenosine at 3' end. 3' end. See, here 3' end have a single adenosine nucleotide. Similarly, here also. This will happen when you amplify the PCR products with the Taq DNA polymerase with a secondary or associated activity. It can add single adenosine at 3' ends of the double-stranded DNA products. See, this is your insert. You want to clone this PCR product into a vector. So we are using TA cloning vector. Now you have to prepare a vector with the overhang of T. This is a circular vector. This circular vector is now linearized by blunt-end DNA restriction endonuclease. Blunt-end restriction endonuclease. One such blunt-end producing restriction endonuclease is SMA1, which is SMA1's restriction site is CCCGGG. When it cuts, one end will have a CCC, other end will have a GGG. So exactly in the center of the restriction site, it cleaves, and you have the vector will have a blunt ends. SMA1 digestion. The SMA1 digestion leads to the linearization of the vector with blunt ends. So such linearized vector is treated with terminal deoxynucleotide transferase in the presence of dideoxythymidine. So now that when you treat with the terminal deoxynucleotide transferase in the presence of this one, it can add terminal deoxynucleotide transferase adds single nucleotide, that's a T thymidine at 3' ends of the vector. You see, the vector having a dideoxythymidine at the 3' end of the vectors. You see, two orangs here and here. So now the insert is having 3' single nucleotide, that's A insert, and the vector having the T. So now insert and the vector are ligated. Then you will get a recombinant plasmid with the insert. This is how you can clone the PCR products. We are exploiting the Taq DNA polymerase activity, that's associated activity. So which is adding the 3' single adenosine. To complementary to this, we are preparing a vector with the T, and both of them are undergoing the ligation to get a recombinant molecule. This is how you can clone the PCR products directly without restriction digestion and all. So this is one of the example of TA cloning vector. That's a PTZ57R by T. You can see here this is a single T and here single T. And the inside having the single A at 3' ends, they are ligated and they are complementary and they'll become recombinant molecules. So at the end of the lecture, I want to ask you one question. Which of the following enzymes adds a single deoxyadenosine to the 3' ends of double-stranded DNA? Here, 4 enzymes. All of them are thermostable enzymes, are used in the PCR. PFU DNA polymerase, very accurate. PFU DNA polymerase, PW DNA polymerase, and PFX DNA polymerase have high accuracy. So, which of the following enzymes adds a single deoxyadenosine to the 3' ends of the double-stranded DNA? That is none other than Taq DNA polymerase. Taq DNA polymerase. This is all about the TA cloning vector. Hope you like my lecture. Thank you for listening. If you want to motivate me and appreciate me, try to subscribe my channel, or you can like it, or you can share it. You can use my material with the free of cost. Thank you all. See you again. Bye-bye.